This is my Microbes and My Food project featuring Pulque and Z Mobilis by James Luna. The Mexica also used to place the image of the dead on those grass wreaths, and the rich sang and drank pulque in honor of these gods and their dead. Pulque is a fermented drink made from agave nectar, originally made by the Mexica, also known as the Aztecs. Drinking pulque was believed to connect people with many gods, especially those connected to fertility in agriculture and humans. Farmers have long cultivated agave, with the fruits of their labor being passed on to noblemen for ritual use. Pulque is fermented by Zymomona mobilis, which metabolizes the agave nectar and produces ethanol and carbon dioxide. Z. mobilis is a rod-shaped facultative anaerobe with a very high ethanol tolerance, making it a threat to commercial brewing operations as it contributes to cider sickness. It is difficult to utilize on a commercial scale due to the limited sugars it can metabolize: glucose, sucrose, and fructose. Pulque is drunk while actively fermenting, similar to kombucha. It occurs naturally in apples and sugary plant saps, including agave and sugar cane. While Z. mobilis can undergo aerobic respiration, the fermentation of agave into a small amount of ATP, NADH+, ethanol, and carbon dioxide make it desirable to humans. While attempts to modify Z. mobilis for alcohol production have largely been unsuccessful, it is useful in producing fuel ethanol. Pulque and Z. mobilis remind us that our cultures and communities are made up of many beings worth honoring.
So today we're going to talk about kimchi and kind of the fermentation behind it. So I'm sure many of you know kimchi is it's a traditional Korean fermented vegetable dish. It's got a rich history. They've been making kimchi for roughly 2,000 years. And it's got a lot of benefits to it. And it's just a great kind of superfood, if you will. So there's a lot of variations of kimchi, but I think most people are familiar with the one that involves Napa cabbage. I think it's kind of the most iconic one. So Napa cabbage is going to be that primary and largest ingredient, accompanied with shredded carrots, spring onions, and then you've got ginger sauce. You've got this flavor sauce, which includes ginger juice, crushed garlic, fish sauce, pureed onion, chili powder, and then so just kind of a little backstory on it. So kimchi was kind of created out of just kind of necessity. Korea was having these harsh winters, and they were trying to find ways to preserve, their food. And while other countries and peoples were, drying their meats or salting their meats, Korea kind of came up with this fermentation technique. And so and that has been going on since kind of the origin of kimchi. So that's been a huge staple for them in their culture. And then once they started industrialization and urbanizing Korea, they started commercialization and started like large scale production of kimchi, which is pretty cool. And then obviously it's widely known around the world, and it's a big export for Korea. So now it's got global popularity, which is amazing, and everyone can know those health benefits that it gives. But so the process of making kimchi, kind of the first thing is brining the vegetables in salt, which kind of draws out all the moisture and inhibits, spoilage bacteria. And they also do thorough cleaning of these vegetables, because if there's any speck dirt or whatever impurity, it can really throw off the fermentation process. And so they make sure they clean it really well just to ensure like a really good batch. And then so kind of the process of it, this was kind of more how they used to do it, and they still do it sometimes now, but the kind of the ancient way of doing it, they have these earthenware pots. And then they kind of pack all the vegetables. Once it's all mixed up, they pack it into their pretty much airtight, and then they put weights on the top to kind of really mash it down. And then that kind of gets all these juices flowing. All these ingredients are kind of creating this oxygen-free zone where all that beneficial bacteria will thrive, while those harmful ones are going to be suppressed by the salt. And then they seal it, and they have this lid that kind of has a water groove. And so they've put water in the groove, and so it prevents air from going in, but allows fermentation gases to escape, which I find fascinating. And then after about a week, kimchi is ready to go. It's fermented. It's got all those that beneficial lactic acid bacteria, which we're going to talk about. Um, so that lactic actic bacteria, or like LAB, that is kind of the primary source of all this for the fermentation, and it's a key player here. So the Leuconostoc is the primary initiator of the fermentation process, particularly Leuconostoc mesenteroids. So those are going to be naturally present on, in the ingredients. They kind of now add them sometimes as kind of like a culture, just kind of maintain safety or just like consistency with their batches, but they generally don't have to add it. It's kind of already there. But so that kind of gets the fermentation process started. And then once there's more acidity in there, the lactobacillus is able to kind of take over and dominate because it can tolerate acidity much more. And that's going to notably be the Lactobacillus plantarum. And then after that, kind of towards the end, the Weissella is going to be the key contributor to kind of giving that complex flavor and that preservation of kimchi. And then, so kimchi's fermentation really relies on that heterofermentative lactic acid bacteria. So it converts these vegetable sugars into lactic acid, ethanol, acetic acid, and carbon dioxide. So this pathway significantly lowers the pH to around 4.2, which inhibits pathogenic microbes and creates that unique flavor compound or compounds. These specialized genes help bacteria maintain internal pH homeostasis in that acidic environment. And then so after, you're eating kimchi, these, or after you kind of make it, these microorganisms survive even after it's prepped. And they can reach and survive the harsh conditions that are in your digestive tract. All that acid, they make their way, all the way down to your gut, colon, things like that. And then that's where they start doing the real magic there is they kind of produce those beneficial bioactive compounds. And those kind of contribute to like your overall health and kind of help give you those benefits. So yeah, some key takeaways here, but yeah, lactic acid bacteria is kind of the main player here that we talked about. And obviously consuming kimchi can give you lots of health benefits, antioxidative and immune stimulatory activities, anti-aging, anti-cancer. It's just a wonderful food. I think everyone should have it in their fridge. All right. Thanks so much for listening.
Hi everyone, today I'm going to talk about natto. Natto is a traditional Japanese food made from fermented soybeans. It smells strong and looks kind of weird, but it's actually very healthy. So, how is it made? First, soybeans are cooked, then a bacterium called Bacillus subtilis natto is added. After that, it's kept in a warm place for about 1 day to ferment. During this time, the bacteria grow and use the soybeans as food. They break down the proteins and other nutrients into smaller molecules, which makes natto easier to digest. The most interesting part is why is natto so sticky and stretchy? It's because the bacteria produce substance called polyglutamic acid. This is a sticky string-like substance that forms around the soybeans. So when you pull it, it stretches it into long strings. Basically, the bacteria are making this sticky material as they grow, and that's what gives natto its unique texture. Also, the bacteria produce enzymes, especially one called nattokinase. This enzyme breaks down proteins into smaller pieces, and it may also help break down blood clots, which can support heart's health. The production of these enzymes is controlled by a gene called APRN, which shows how genes and metabolisms are connected. Another cool thing is that some of these bacteria are still alive when we eat natto, so they can act like probiotics, which may help to support our gut. So, in this process, the bacteria benefit because they get nutrients from the soybeans, and we benefit because the food becomes more nutritious, easier to digest, and even has extra health benefit. Thank you for watching.
Say hello to Leuconostoc mesenteroides, a microbiological look at kimchi. So, looking at our microorganism, Leuconostoc mesenteroides, a few things to know about it. It does appear in the coccus and streptococci arrangements, though it can vary a little bit depending on the colony that you're looking at and the type of media that you're looking at. So, in a solid media or a media that has glucose in it, it sometimes can appear a little bit more elongated. It is a facultative anaerobe, meaning it can undergo aerobic respiration in the presence of oxygen or fermentation and anaerobic respiration when it is not in the presence of oxygen. It also stains gram positive and has a low GNC content. Now, as I mentioned, it can undergo fermentation. That's a huge role in making kimchi. Now, how this is done is Leuconostoc mesenteroides is actually a naturally occurring microorganism. So it starts in that cabbage and really just gets activated in the presence of moisture. So brining it in some salt water or using a dry salt to bring out moisture from the cabbage, along with any flavoring ingredients that you want to include in there, is really what activates it. And then if you close off its access to oxygen by putting it in a jar, you can allow that process of fermentation to happen. Now, it's a relatively quick process for this microorganism. It's usually complete in about 1 or 2 days at room temperature, or 3 or 4 days when refrigerated. Now, a fun fact to leave you with is that kimchi's actually been into space, but Leuconostoc mesenteroides has not. So in 2008, when South Korea sent their first astronaut into space, they sent them up with a can of kimchi that had been treated with radiation as to kill all the bacteria in it and make sure it was safe to be up in space.
Hi, my name is Steven, and I research Lactobacillus sakei, which can be found in kimchi, a fermented vegetable or root with origins in Korea. Historically, kimchi began as a simple salting of vegetables, and which drew out moisture to preserve them. And the kimchi we know today has added many ingredients to make the unique taste, like red pepper powder, garlic, ginger, and green onions. The process of making kimchi is culturally significant, where families or communities will gather together in the wintertime and prepare a bunch of kimchis and share with everybody, and that's called kimjang. Lactobacillus sakei is a rod-shaped lactic acid bacterium. It's gram-positive and doesn't form any endospores. It thrives in environments with high concentration of NaCl, temperatures ranging from 4 degrees Celsius to 48 degrees Celsius, and it's anaerobic. And this is very significant because, as I mentioned earlier, kimchi is made and then put in the fridge, and that's the perfect environment for Lactobacillus sakei to live in. There are 2 types of lactic acid fermentation in kimchi, but Lactobacillus sakei is homolactic fermentation. So you start off with the glycolysis, the breakdown of the sugars usually found in vegetables, the substrate being glucose, and the products are 2-pyruvate, 2-ATP, and 2-NADH. After glycolysis is homolactic fermentation, when the pyruvate turns into the lactic acid, which allows NAD plus to be recycled. Lactic acid helps preserve the kimchi because it lowers the pH, making it sour. Some of the relevant genes are the glycolysis genes PFK, PYK, LDH, XPK, and there are no genetic modifications that are used in kimchi since the process is all natural.
Okay, so I did my project on the wild yeast species. This is my sourdough starter. She's been mixed a few times, so she's not showing the appropriate gas bubbles anymore. Maybe if I get further down. There's a couple down there still. Sorry, I've had to take a couple of takes on the video, so she's a little gassed out now. But anyways, while we look at her, I will just briefly go what yeast is all about. So let's see, Candida milleri is also synonymous with Malthyma humilis. It's one of the most representative yeast species found in sourdough. It originated over 4,000 years ago in ancient Egypt, making it one of the oldest forms of leavened bread. Sometime around 3,000 to 1500 BCE, microscopic analysis of recovered dissected bread loaves revealed this yeast species. One baker back then noticed the warm climate allowed flour and water to ferment as natural yeast and bacteria colonized the dough. They realized that made bread rise and taste better, creating the sourdough process. Flour naturally carries wild yeast and lactic acid bacteria on its surface, so mixed with water and allow time and warmth, the microorganisms start feeding on the starches, producing carbon dioxide and organic acids, giving rise and tang for the flavor. Almost immediately, the bacteria starts feeding on the sugars from flour and water, and after 3 days, lactic acid bacteria are made associated with no other bacteria can thrive. By day 14, you have what you're looking at here, and you're ready to make sourdough bread. Fun fact: during the first March of COVID, sourdough interest actually spiked 500%. So amphimalis is slower fermentation, supports the lactic acids like lactobacillus sanfrancisnus, which can have many health benefits such as mineral production and lower glycemic index for our bodies. It's highly air tolerant and thrives in high acidic and high salt environments. Its ability to metabolize fructose, glucose, lactose, triglyceride, and glycerol, but not maltose, which is great because its partner, lactic acid, loves maltose. So they are more neutral partners, not synergistic. Once your dough is cultivated with and you got your sourdough starter, you put warm water, your starter, flour, salt, and butter if you like, and then you mix and you for approximately 8 minutes, letting it rise. During the first bulk rise, that's when fermentation happens. So your dough will rise with all of the gas, all of the CO2 that is produced, and then you will let it proof, which does a more a taste. It gives the tang more for 12 hours on the counter or in the fridge, and then your final step is baking, to which then you will have nice pocketed holes inside your bread, giving the swish, and that is due to the carbon dioxide produced from amphimalis. Let's see, the last thing is the metabolic pathway or glycolysis. When the starter is first made, yeast thrives on the glucose, consuming it and turning it into ethanol and carbon dioxide. Wild strains such as amphimalis are known for varied and efficient carbon use, including consuming the organic acid and alcohol. There's also another type of metabolism called the dioxide shift, where after the glucose is consumed anaerobically, even with the oxygen present, producing the ethanol, and once glucose is depleted, the yeast enters the dioxide shift, changing their metabolism to consume the ethanol that they previously produced through efficient aerobic respiration. And they have a very significant genetic diversity, allowing adaptation to different environments, thus why they can survive and thrive in here. And one last look, we'll try to find those carbon dioxide bubbles. You can see them a little bit as I let her sit here for a moment. But yes, she's very hungry now that I have taken all of her CO2 out. I should probably put some flour. Anywho, thank you for listening. That is all.
Hey guys, so today we're going to be going over Lactobacillus crispatus, which is a microorganism that can be found in the vaginal microbiota. But before we get into that, I do want to go over a few background things. The human microbiota is a complex and dynamic ecosystem that contains trillions of organisms. Fun fact for you, there's about 3 million expressed bacterial genes in the gut microbiota, but only 20,000 expressed in the human genome. Products of the human microbiota assist in metabolic regulation, barrier function, immune system homeostasis, pathogen invasion prevention, and organ signaling. And even though the microbiome is complex and dynamic, it can be disrupted. Dysbiosis is the disturbance of microbiota homeostasis. And generally, there's 3 different types. You've got loss of beneficial organisms or microbial diversity, and or excessive growth of potentially harmful organisms. Things like antibiotics, diet, hormonal changes, and hygiene practices can all disrupt microbial homeostasis. Dysbiosis can generally lead to chronic inflammation, a weakened intestinal barrier, a weakened immune system, and even contribute to health conditions like obesity and diabetes. The vaginal microbiome is unique because it doesn't favor microbial diversity. It's actually lactobacillus dominant. The ecosystem is known to be high in glycogen and mucin, has a low pH, lacks oxygen, and tends to shift and change with hormonal fluctuations. Vaginal microbial communities that are more diverse have been linked to bacterial vaginosis, sexually transmitted diseases, miscarriages, and preterm birth. The vaginal microbiome is categorized by 5 different cervical types. CST1 is Lactobacillus crispatus dominant. CST2 is Lactobacillus gyseri dominant, so on and so forth. It's important to note that cervicotypes that are Lactobacillus dominant are considered to be the hallmark of vaginal health. CST4 are generally more at risk for all things listed on the previous slide, from BV to STDs, etc. Lactobacillus crispatus are gram-positive, non-spore-forming bacteria. They're rod-shaped and considered aerotolerant anaerobes. They metabolize glucose and lactose through fermentation because the vagina is a low-oxygen environment. Lactobacillus crispatus primarily produces lactic acid as its fermentation end product, but it can also produce hydrogen peroxide and bacteriocins. Lactic acid helps maintain its acidic environment, which basically stops bacterial growth. Bacteriocins help stop infection before it starts by preventing specific microbes from binding to the vaginal epithelium. Biosurfactants, also produced by Lactobacillus crispatus, also helps prevent the binding of specific microbes to the epithelium and prevents biofilm formation from harmful bacteria. The Lactobacillus crispatus' multifaceted defense mechanism makes it especially unique. These characteristics are important in the vaginal microbiota because it all helps maintain a stable ecosystem that's resistant to disruption.
Hi, this is Neon Brooks, and I'm going to be giving an overview of Escherichia coli and the human intestinal microbiome. The human microbiome consists of the complex microbial communities on our skin, our digestive and reproductive tracts, and elsewhere in our bodies, which co-evolved with our species. This includes many genuses of bacteria, as well as archaea, fungi, and even eukaryotes and viruses. The health and makeup of our microbiome is dependent on numerous factors, including our environment, social and animal interactions, diet, physical activity, and medications, including antibiotics. The intestines is one of the most densely packed ecosystems on the planet, containing many microbes that play important roles in digestion, health, and disease. The intestines are a very oxygen-poor habitat, and most bacteria in this ecosystem are anaerobes. Or are strict anaerobes. pH is typically between 6.0 and 7.5, and the environment is very wet in the duodenum and dries out as fecal matter moves through the tract towards the rectum. The digestive tract is highly metabolically rich given the presence of food matter that is undergoing digestion. The intestinal microbiome is highly diverse and can vary considerably across individuals, with bacteria from 5 or more distinct genera of bacteria typically present. Microbiota assist the host through the extraction, absorption, and even synthesis of nutrients and metabolites. They also protect the intestines from pathogenic bacteria colonization and regulate immune cells. Gut microbes compete with one another for space and resources, but they also cooperate, such as when different bacteria exchange metabolites in mutually beneficial ways. Escherichia coli is a common bacterial resident of the intestines. It's a gram-negative, rod-shaped bacterium that has both fimbriae and flagella. E. coli is a form of gamma proteobacteria. Within this category, E. coli is one of many glycolytic facultative anaerobes, which catabolize carbohydrates through glycolysis. There are many independent strands of E. coli. Most are harmless or beneficial to the gut, but can still serve as opportunistic pathogens, but some strains are highly pathogenic. E. coli confers several benefits to its human host as part of a healthy intestinal community. As facultative anaerobes, E. coli can consume oxygen that's toxic to the many strict anaerobes in the microbial community. They also produce vitamin K and vitamin B12. And lastly, one of their most important contributions is simply to take up real estate and thus outcompete potentially harmful pathogens.
The human microbiome is the who, what, where, why, and what of what is living inside of us. So we have a microbiome that lives and covers our body. It has things like bacteria, fungi, and viruses that inhabit our entire body. It's a symbiotic relationship. I'm going to be talking about the vibrant microbial community that's essential in maintaining a healthy reproductive and urinary system. So the combination of healthy microbes and the constant shedding of the stratified squamous epithelial cells create a self-cleaning environment for the vagina. The vaginal ecosystem is dark, moist, and warm. It naturally has low oxygen, low pH levels, which creates a perfect home and nutrient center for many different microbial species. Your reproductive age, hormone levels, and other physiological changes cause fluctuations in the microbiome.
Hello, my name is Aaron Kelly, and I chose to do my essay on the microbiome of the gut. And I learned that humans have a symbiotic relationship with thousands of species of microbes that live on the skin, in the gut, and throughout our bodies. According to the Human Microbiome Project, our microbiota is responsible for assisting with digestion, producing essential vitamins, and helping with our immune response. The gut is the largest habitat for microbes in the human body, and the gut microbiome is the most studied part of the human microbiome. Studies have shown that an active lifestyle and diets, such as the Mediterranean diet, correlate with a more diverse gut microbiome and fewer microbes that are known to cause inflammation. Fortunately, that means that we can make healthy lifestyle choices to improve our gut health. Unfortunately, being exposed to certain chemicals or using antibiotics can have a negative impact on our microbiome by killing the healthy microbes that live in our gut. This can lead to an imbalance of microbes in the gut, which can mean either an overpopulation of harmful bacteria or a reduced population of helpful microbes. There is growing evidence that microbial imbalances are linked to many diseases, including type 1 diabetes and other autoimmune diseases. A common type of microbe found in our gut is the Bacteroides. They usually live happily in the gut, breaking down complex molecules for the host, but can occasionally become opportunistic pathogens, causing illness in immunocompromised individuals. Scientists are still learning about our microbiome and how it interacts with the rest of our body. I can't wait to see where the future research leads us.
Okay, guys, this might sound weird, but bacteria in your gut can actually affect your mood. So, the one I'm talking about is called Bifidobacterium. It's a beneficial bacteria that naturally live in your gut, especially in your large intestine. People call it good bacteria because it helps keep your gut balanced. It helps break down part of food your body can't fully digest on its own. And it's also produce substance that help keep your gut lining healthy and support your immune system. It's also protect your gut by stopping harmful bacteria from growing too much. But the most interesting part is, it's not just about digestion. Your gut and your brain are connected to something called the gut-brain axis. This bacteria can influence stress, mood, and chemicals like serotonin. When it balances, it supports your mood, but when it's not, it can affect how you feel. So, something really small like bacteria can have a big impact on how you feel every day. And that's why taking care of your gut also means taking care of your mental health. Goodbye!
Hello everyone, this is my presentation for the Microbes in Me homework assignment. What is the human microbiota? This is a collection of all microbes that live in and on the human body, things like bacteria, viruses, fungus. We have quite a lot of them and they are found everywhere. These are some examples of microbiomes that we have within us, like on our skin, gastrointestinal tract, and reproductive systems. Things that can harm our microbiota are antibiotics, although they can be good for getting rid of bad bacteria. Sometimes they can also end up harming good bacteria that we have and that we need. Diet is a really big important thing and impact on our microbiota. Skin products and hygiene products also can cause harm. The microbiome of the urinary system is fairly new in research. However, they have found differences between men and women, mostly due to hormones and anatomical features. When there are imbalances in this system, it can cause infections and other diseases. Citrobacter frundi is a bacteria that's facultative. It belongs to the Enterobacteriaceae family and its genus Citrobacter. It is gram-negative and non-sporing. There are around 11 species within this genus, and most of them are found in the gastrointestinal tract of animals and humans. The impact of C. frundi in the urinary system. In recent times, it has been the cause for urinary tract infections in hospital patients who have been there for a long time. Usually caused because of the use of catheters. This bacteria is very interesting because it has become resistant to multiple antibiotics, and this is partly due to
is plasmids, because they are able to adapt very quickly to their environments. And apart from the urinary tract infections that they can cause, they can also cause other diseases like meningitis, pneumonia, diarrhea, and sepsis.
Hello everyone. For my homework assignment, I have selected Microbes in Me, which will explore Faecalibacterium. First, a quick overview. The human body has many sites that don't have microbes, but the sites that do house microbes have trillions. The human body and the microbe relationship is normally commensal, meaning the microbes benefit from the human body but do not harm us. There are, however, mutual benefits, such as microbes helping the human body digest dietary fibers and supporting with 10% of our caloric intake. Microbial communities can be disrupted, and this can lead to negative outcomes. An example is antibiotic treatment. The antibiotic does not target a specific pathogen, so this often leads to collateral damage, impacting other bacteria. I focused on the large intestine, which has a pH range of 6 to 7, so it's slightly acidic and neutral. It is an anaerobic environment. It has an abundance of metabolites. 70 to 90% of the bacteria in a usually healthy gut are Bacteroides and Firmicutes, but other bacteria are also present. Bacteroides and Bifidobacterium can help break down complex plant cell wall polysaccharides, and the byproducts are used by the human body. Going back to the antibiotic treatment, so this can disrupt the human microbiota. In a healthy gut, there are microbes that support a balanced immune response. And so a decrease in this population can allow pathogenic bacteria to proliferate and trigger pro-inflammatory responses. Faecalibacterium, the focus of this assignment, contributes to a healthy gut. It belongs to the phylum Firmicutes, and only 6 species have been validly named. It is strictly anaerobic, gram-positive, rod-shaped, non-motile, and non-spore-forming. Faecalibacterium is associated with anti-inflammatory mechanisms by blocking proteins that activate when there are pro-inflammatory stimuli. Additionally, another benefit of Faecalibacterium is that it produces the metabolite butyrate, which can strengthen the gut barrier.
My presentation is on kombucha and Comagudibacter xylanus. Kombucha is a slightly acidic carbonated beverage created by fermenting tea. The first drink was utilized by Northeast China in 220 BC for medicinal purposes. The tea, sugar, and a culture called a SCOBY are the core ingredients of kombucha. A SCOBY is a symbiotic colony of bacteria and yeast. It is mainly composed of acetic acid bacteria, yeast, and small amounts of lactic acid bacteria. The SCOBY not only ferments the beverage, but also provides an array of probiotics, which can promote gut health. The fermentation process used to brew kombucha produces trace amounts of alcohol. However, kombucha is still considered a non-alcoholic beverage by the FDA. A kombucha SCOBY largely consists of a mix of acetic acid bacteria and yeast. In smaller scale production, once the tea is prepared for fermentation, a SCOBY is placed on the liquid surface. Then the container is covered with a breathable cloth and left for 7 to 21 days. The production of this cellulosic biofilm floating on the surface of the tea broth in kombucha is associated with the presence of the AAB species Comagudibacter xylanus. This acetic acid bacteria species is a gram-negative, catalase positive, and rod-shaped obligate aerobe. X. xylanus is also characterized by producing cellulose, a thick organic material that grows on a liquid surface, one of the main components of a SCOBY structure. During fermentation, a newly formed jelly-like daughter tea fungus membrane is formed, which floats on the surface of the broth. The process described is a direct result of K. xylanus producing a new layer of cellulose in addition to the SCOBY, repeating every kombucha fermentation cycle. Once the liquid has fermented and the SCOBY is removed, the drink still contains living organisms. In a collaboration between Eugen Drinks and the University of Ghent, it was discovered that every bottle or can contains as much as 3 billion living microorganisms. Fermentation plays a key role in creating kombucha. It develops the flavor, carbonation, pH, and life inside the drink. Black tea is the most common tea substrate for kombucha, and sucrose is the most popular source of carbon during fermentation. However, other teas can be used as well as various herbs and fruits to aid the process. After the tea, sugar, SCOBY, and any other substrates have been sealed, it's time for fermentation to take place. The yeast from the SCOBY performs a smaller scale fermentation called ethanol fermentation. The sugar and fruits added provide sucrose for the yeast to hydrolyze, breaking sucrose down into fructose and glucose. Next, the yeast cells break down the monosaccharides into ethanol, which marks the beginning of acetic acid fermentation. Acetic acid bacteria from the SCOBY oxidizes the ethanol, creating acetic acid. This acid is more commonly recognized as vinegar, which can explain kombucha's unique taste. Once fermentation is complete, the original and daughter SCOBYs are removed from the kombucha to be reused. The tea is filtered to remove impurities and transferred to a sealed bottle in a cool environment. This is because if raw kombucha is kept unrefrigerated, the beverage is able to continue its own fermentation.
We as people like to think of ourselves as individuals. In reality, we are each a whole world. With our own diverse ecosystems, there are ecosystems in our hair, our mouths, under our nails, and on the bottoms of our feet. We have enough life to fill a solar system in just the 25 feet of our intestines. We have those that are our friends, like the demodex that live in our follicles and help keep us clean, or the microbes that live in our guts to help us process what we can't by ourselves. Then there are the foes, the ones that make us sick, the ones that make us itch or hurt or bleed. Staphylococcus aureus is a gram-positive spherical bacterium that typically forms clusters and is commonly found on the skin and in the nasal passages of a healthy individual. Although it is often part of the normal microbiota, it can act as an opportunistic pathogen when it enters the body through breaks in the skin or when the immune system is compromised. This organism is capable of causing a wide range of infections, from minor skin conditions like boils and abscesses to more severe diseases such as pneumonia, bloodstream infections, and toxic shock syndrome. Its ability to cause disease is linked to various virulence factors, including toxins and enzymes that help it invade tissues and invade host immune defenses. Additionally, some strains such as methicillin-resistant Staphylococcus aureus have developed resistance to multiple antibiotics, making infections more difficult to treat and a significant concern in healthcare settings. Staphylococcus aureus plays a major role in the progression and severity of atopic dermatitis and a smaller secondary role in seborrheic dermatitis. In atopic dermatitis, the skin barrier is weakened, which allows the bacterium to colonize the skin more easily and persist in high numbers. Once established, S. aureus releases virulence factors such as toxins and superantigens that overstimulate the immune system and promote strong inflammatory responses. This leads to worsening redness, itching, and chronic flare-ups typical of atopic dermatitis.
Haemophilus influenzae is a significant bacterial pathogen that frequently causes eye infections, particularly in children. Despite its name, it does not cause the flu, but it is a common cause of bacterial conjunctivitis, also known as pink eye. H. influenzae is characterized as a small 0.3 to 1 micrometer gram-negative coccobacillus. Under a microscope, it appears red and pink after gram staining and is phelomorphic, meaning its shape can vary between a short rod and a coccus. As a facultative anaerobe, it grows best at 35 to 37 degrees Celsius and requires specific factors present in chocolate agar for cultivation. These bacteria are non-motile and commonly possess specialized villi for adhesion to mucous membranes, allowing them to resist removal by blinking or tears. In the eye, H. influenzae usually cause causes non-invasive mucosal infections, particularly in the conjunctivitis. The symptoms are intense eye redness, swelling, burning, intense itchiness, and a thick yellow or green purulent discharge that can crust the eyelids and shut overnight. It spreads easily via direct contact with respiratory secretions and hand-to-hand contact or droplets. It's highly common in daycare settings and often occurs with an ear infection, and often in children. While rare, it can cause severe ocular conditions like keratitis and endophthalmitis, especially in immunocompromised individuals. Clinical diagnosis is usually made by identifying the purulent conjunctivitis and confirmed by swab cultures taken from the lower palpebral conjunctiva. While mild cases may resolve, antibiotic drops or ointments can be used to shorten the duration, typically lasting 7 to 10 days with treatment. Many strains produced beta-lactamase, leading to resistance against ampicillin, thus requiring careful antibiotic selection. H. influenzae remains a predominant cause of pediatric conjunctivitis that requires prompt hygienic measures and often antibiotic treatment to resolve your symptoms.
I chose to focus on the skin microbiome and specifically Cutibacterium acnes. I chose this topic because I work in a dermatology clinic where we treat many patients with acne with various active medications depending on the severity of their condition. And I was curious to learn more about the communities that exist on the skin and what causes acne. Zooming out a little bit, our body is home to trillions of microorganisms. For bacteria alone, it is estimated that there are around 38 trillion bacteria living on our body. That's more bacteria than there are cells making up the human body. And then going to the skin specifically, it's an incredibly complex and diverse community of microorganisms, as shown in this image here. And even within the skin, the various locations, depending on the density of different glands, differs significantly. Overall, the skin is relatively dry, acidic compared to other ecosystems on the human body. One important player within the ecosystem of the skin is Cutibacterium acnes, which is a gram-positive, aerotolerant, anaerobe that lives in hair follicles. It is associated with acne, but under normal conditions, it is completely harmless. It is even beneficial, helping regulate the population of other pathogenic organisms. But certain strains of C. acnes contain virulent genes that can trigger inflammation. I found it interesting that acne isn't necessarily associated with having more of these bacteria in sebaceous glands, but it is associated with the higher density of strains that are associated with acne and are more often pathogenic. I think that understanding the specific different strains of C. acnes can help us develop therapies that might target these strains, one or multiple specific strains, and more effectively treat the disease.